The cerebellum, unique organization from cerebral cortex, the cerebellar hemispheres, vermis. You have zones, fissures, folia, lobules, deep nuclei, and peduncles. Has a unique cellular makeup from cortex, has unique neurons, mossy fibers, climbing fibers, Purkinje cells, and granule cells. It inputs to and outputs from the cerebellum. Function linked to its parts and zones, so movement, learning, cognition, etc. And we'll talk about damage to the cerebellum. So the cerebellum is the little brain. In some ways, it's like a whole other brain. Only 10% of the entire brain. But it has more neurons than all the rest of the central nervous system combined. It is densely folded, and if it were spread out, it would be 1 meter long, 10 centimeters wide. It is more folded than the gyri of the cerebral cortex. The cerebellum is a workhorse. It is the most underrated part of the brain, in my opinion. Metabolically, it is one of the most active brain parts. Has unique cellular structure, not 6 layers, but 3. It is homogeneous tissue, so there's no cytoarchitectonic based regions like the cerebrum. All sensory info passes through the cerebellum except olfaction. It's also indirect part of the motor system, so damage to the cerebellum equals motoric problems. The cerebellum functions largely outside of conscious awareness, so tuning, timing, adjusting, posture, and movements and balance. So we'll look at the gross anatomy. There are 2 cerebellar hemispheres, bilateral structure left and right. There's a gray matter cortex, underlying white matter, banded or striped gray matter layers or folia, deep fissures divided into lobes. So you have an anterior lobe and a posterior lobe. The left and right pairs of peduncles are superior, middle, inferior, and connect the cerebellum to the brainstem. The vermis is the midline structure. And you have deep cerebellar nuclei nestled inside of the white matter. So this is your fastigial, interpositus, emboliform, globose, and dentate nuclei. So those nuclei are important. Fastigial, interpositus, emboliform, globose, and dentate nuclei. So the general anatomy of the cerebellum is here. You can see as we pivot around, the cerebellum has 2 hemispheres. The vermis or worm is in the middle, and it is attached to the brainstem through those peduncles. All right. So you have the 4th ventricle, which is open to the cerebellum. You have a flocculus, a tonsil, pyramid, middle cerebellar peduncle, inferior cerebellar peduncle, and superior cerebellar peduncle. So the peduncles are important to note. So here are major parts in multiple views. You need to be acquainted with the orientation of the cerebellum. So you have cerebellar hemispheres, vermis in the center, and then fissures. So it's important to be able to distinguish the orientation. You have a primary fissure, which separates into anterior and posterior lobes. You have a horizontal fissure, which just separates posterior lobe top from bottom. There are lobules of vermis and beyond. We have the supplementary medullary velum, the central lobe, the culmen, the primary fissure, the declave, the folium, the tuber, the pyramis, which I think is supposed to be pyramids, uvula, nodulus, tonsils, hemisphere, and then anterior lobe and posterior lobe. So anterior lobe is A, B is posterior lobe, and then just be able to recognize some of the most important features like the tonsils, let's see, the nodulus, the folium, declave, and definitely know the primary fissure and central lobe. There's a lot of important vocabulary here. So the vermis has a Roman numeral system, Roman numeral system. So 1 and 2 are the lingua. Number 3 is central. Number 4 is culmen. Number 5 is culmen. Number 6 is declave. Number 7 is folium and tuber. Number 8 is pyramis. Number 9 is uvula. And number 10 is nodulus. So there's a lot of terminology associated with the cerebellum. Some may say too much terminology. You have the Roman numeral system, the classic names, the newest nomenclature, and then there are other names. So just be aware of this terminology for the cerebellum. If you see lingula, central lobule, culmen, declave, upper part folium, lower part tubule, pyramis, uvula, and nodulus. So the white matter, you can see the arbor vitae, or tree of life. The cerebellum has white matter at its center, just like the cortex does. It is called the arbor vitae, or tree of life. Deep in this white matter is also housed the deep cerebellar nuclei. There's a stained picture above so we can well see the dentate nucleus, which is teeth-like. So this stain here is the dentate nucleus. Deep cerebellar nuclei. So there are 3 pairs of cerebellar peduncles, superior, middle, inferior, and they are very different white matter fibers carrying very different types of information. So you can see the superior cerebellar peduncle attaches the cerebellum to the midbrain. The middle cerebellar peduncle attaches the cerebellar peduncle to the pons. The ICP, the inferior cerebellar peduncle, attaches the cerebellum to the medulla. And the tractography, the orange fibers are coming in, so that's input via the ICP from the olives. Red are the fibers coming in from the input via the MCP from pons, which come from the cortex. And the purple fibers are leaving, or the output, the cerebellum via the superior cerebellar peduncle to the thalamus. So the VL to loop back to the cortex. So superior cerebellar peduncle, you can see them underneath the colliculus, and so they're attached at the pons. So you have superior attached at midbrain, middle attached to the pons, inferior attached to the medulla. Here we have the longitudinal zones. We have the vermis, the medial hemisphere, the lateral hemisphere, but also horizontal segments such as the flocculonodular lobe. So it's important to be able to recognize these longitudinal zones. So looking at the vermis, the medial hemisphere, and the lateral hemisphere. And then you can also see the different lobes. So you have the anterior lobe and the posterior lobe. And floccular lobe. All right, so this is important. This is the deep cerebellar nuclei. You can see the globose and emboliform nuclei, the dentate nucleus, and the fastigial nucleus. This is a good picture here. You have the fastigial nucleus in the middle, the globose and emboliform nuclei, which are going to be a little bit lateral of the midline there. And then the dentate nucleus, which it's a teeth-like dentate, you can see stained there, it's all crinkly. So dentate nucleus, crinkly. And then as you get into the middle, you have the interposed nucleus, the globose and emboliform nuclei. And then towards the middle, you have the fastigial nucleus. So we have longitudinal zones and deep nuclei. The vermal zone projects to the fastigial nucleus, so that you have the intermediate or medial zone, which projects to the interpositus nucleus, and the lateral zone, which projects to the dentate nucleus. So you can follow along here, and we'll see this section. So you have the vermal zone in the middle, intermediate or medial zone, which projects to the interpositus nucleus, and the lateral zone, which projects to the dentate nucleus. So this is allowing you to see the deep cerebellar nuclei and the zones in which they project to. So vermal zone goes with fastigial nucleus, which makes sense because the vermis is in the middle. The fastigial nucleus is in the middle. And then the intermediate or medial zone goes with the interpositus nucleus, which we know is that weird globulous name. Has two names. And then the lateral zone goes with the dentate nucleus. So just think of it sort of as it's topographically arranged, or like the middle goes with the middle, medial, medial, and then lateral with lateral. Here's another picture. This is the longitudinal zones and the deep nuclei again. You can see the deep nuclei here in a cartoon. The vermal zone projects to the fastigial nucleus. The intermediate or medial zone goes to the interpositus or interposed nucleus, and the lateral zone projects to the dentate nucleus. All right, so here we have this cellular organization. This is really important. So instead of 6 layers like the rest of the cortex, the cerebellum has only 3. You have the molecular outer layer, where there's no neurons, but mostly synapses. You have the Purkinje middle layer, which contains Purkinje cells, shown here in the blue arrows, and single-layer large cells. Then you have the granular inner layer next to the white matter. These are many tiny, tightly packed granule cells. So it's important to know molecular outer layer, Purkinje middle layer, and granular layer is the inner layer next to the white matter. So the flow here, you're going to have input to the cerebellar cortex, then the deep nuclei, and then the output to the target. So it's going to go, like here you can see input from the lateral cerebellar cortex, and then it goes to the dentate nucleus, and then it's going to go to the output target. So inputs to the cerebellum are excitatory, and output from the deep nuclei are excitatory. The cerebellar cortex to deep nuclei are inhibitory. They are the axons of the Purkinje cells. So coming in, it's excitatory, and the output from the deep nuclei are excitatory, but from the cerebellar cortex to the deep nuclei, it's inhibitory. So excitatory, then coming in to the cerebellum, and then you have the cerebellar cortex, which is going to project like the dentate nucleus here, and that's going to be inhibitory to the dentate nucleus, and then it's going to be excitatory to its output target. So there are major cell types like climbing fibers and Purkinje cells. The Purkinje cells, the only axons that leave the cerebellum. So Purkinje cells have axons that leave the cerebellum. Then you have climbing fibers, which climb up and wrap around the Purkinje cells to create thousands of synapses. And then you have mossy fibers, which end on the dendrites of the granule cells, which then directly project to Purkinje cells. So know these major cell types: Purkinje cells, climbing fibers, and mossy fibers. So here we have some of the connections. So you can see the typical connection, or at least a simple one, where you have a principal circuit through the cerebellar cortex. You have climbing fibers that wrap around Purkinje cells and make direct connections. Then you have mossy fibers, which synapse first on the granule cells, and then via parallel fibers that synapse on Purkinje cells indirectly. So we have red inhibitory and green excitatory. So the P, I'm guessing, are the Purkinje, and you have the climbing fibers, which are going to wrap around the Purkinje cells, and then you have the mossy fibers, which synapse on the granule cells, and then via parallel fibers synapse on the Purkinje cells indirectly. So we have the cerebellar structure and function. Cerebellum is involved in equilibrium, control of muscle tone and posture, coordination of voluntary movements. Thus, the cerebellum receives inputs from vestibular, spinal cord, and cerebral cortex areas. There are different cerebellar parts that have different functions. The flocculonodular lobe and the uvula receive vestibular input to fastigial nucleus. The vermis and the medial zone receive spinal inputs to interpositus nucleus. The lateral hemispheres receive input from the cerebral cortex through the pons to the dentate nucleus. Vestibular inputs mostly reach the flocculus and nodulus, but also the vermis. They coordinate posture and balance, and also the flocculus is important in eye movements, which obviously visual information is important for balance. So you can see the anatomy here. You can see the lobes, the posterior lobe showing the vermis, and the anterior lobe showing the vermis, the primary fissure, which is going to separate the anterior and posterior lobes. And then you can see the two hemispheres, and some notable features are the flocculus and the tonsil. You also have a horizontal fissure, which is important to see. All right. So somatosensory input from spinal cord. Somatosensory info from mechanicoreceptors reaches the vermis and medial hemispheres. These projections retain somatotopic organization, not as precisely as other parts of the brain, though, but mostly ipsilateral. So you have areas of increased blood flow during repeated flexion and extension of the ipsilateral hand. So you can see this here on the fMRI study in red and orange, or the foot, which is in blue. So you have hand movement that activated multiple small areas, all of them separate from the area activated by foot movements. So in C through E, you can see the average areas of maximum activation in 46 subjects as they moved various body parts seen from above, behind, and below. So cortex to cerebellum via the pons. The cerebellum also receives lots of input from the cerebral cortex, so pons to MCP. It is the least well understood. It is a very promising area of discovery in cogneuro. The lateral hemisphere is mostly involved in cognition, for example, planning of movements, working memory, and motor learning. Through pons to cortex from cerebellum, the rhesus monkey. Distribution of projections derived from association cortices in the prefrontal or purple, posterior parietal in blue, and temporal red, and the parastriate and parahippocampal regions in orange, and from motor, premotor, and supplementary motor areas in green. Timing of movements. fMRI studies show that lateral cerebellar hemispheres activate just after the movement planning regions that activate premotor, SMA, but before the movement happens. So the lateral cerebellar also activates just after movements begin, which is consistent with adjustment and timing of the movement. So damage to the cerebellum. If you have general damage to the lobes or zones, it has a predictable outcome. So midline damage, you're going to have problems with posture, for example, swaying. This is also seen in advanced alcoholics. Lateral damage, you're going to see ataxia of limbs, muscle tone, reflex, coordination problems, for example, staggering. And then flocculus damage, you'll have eye movement problems. So there are some key points about damage.